Hi, I'm Elena Westby, and I'm going to be talking about the time series analysis of the first 2 hours of the Italian honeybee immune response. So a little bit of background on Italian honeybees. They are the most widely distributed insect pollinator. So they are found on all of the continents excluding Antarctica, and they are the most frequent pollinators of the widest range of plants, making them the most important insect pollinator of all of them. And so of the 100 crop species that make up our global agriculture and all of our food that we eat, bees pollinate nearly 70% of them, meaning our global agriculture is heavily reliant and dependent on bees as pollinators. So to kind of put this in perspective, 1/3 of the food on our plate is entirely dependent on honeybees. So if they were to disappear, we would lose an entire third of the food that we eat. And just to kind of reiterate how important pollination is by honeybees, commercial orchards will employ beekeepers to bring colonies to their orchards to kind of help pollinate. And as a result of that, they saw a 20% increase in yield, which essentially means that they were bigger, better quality, just kind of emphasizing how important they are to us. And then this is a figure that kind of demonstrates the multiple ways that we are in fact heavily reliant on bees. So with our food, our crops, trees, honey, which is used in various industries. And so as a result of that, they have a very large economic impact on our society. So this figure is kind of showing what crops are the most dependent on honeybees. So say, for example, we were to lose all of our honeybees, we would no longer have almonds or cauliflower or celery. So we, as a society, are extremely dependent on them. This is important because in the winter of 2006, beekeepers reported massive losses in their colonies. So as we would have had maybe 17% loss, we jumped immediately to 37%, which is concerning because we are so reliant on them. And 50% of these lost colonies demonstrated symptoms that are not correlated to any other known cause of honeybee death. So this phenomenon has been termed colony collapse disorder. And what this looks like is a complete loss of all of the worker bees in the colonies. So that leaves only the queen and the young bees, which cannot sustain the colony by themselves, causing it to die. So the interesting thing about colony collapse disorder is that all of the worker bees have disappeared, but there are no dead bodies found anywhere near the colony. So as a result of this, no scientific cause has been identified for colony collapse disorder, but research points to it being a synergistic effect of multiple factors such as climate change, transportation, pesticides, and most notably to us, parasites and disease, which are of pathogenic nature. So that means that if we investigate their immune system, we can kind of gauge how we can effectively mitigate this. So seeing as bees are invertebrates, they have innate immunity as opposed to adaptive immunity. And what that means is they have three main paths of attack in terms of an infection. And those are antimicrobial peptides, phagocytosis, and encapsulation through the melanin synthesis cascade. And so the melanin synthesis cascade is an evolutionarily conserved immune pathway, and it involves the cleaving of prophenyl oxidase into phenyl oxidase to produce melanin, which then surrounds a pathogen and encapsulates it, essentially quarantining it and allowing it to, not allowing it to have any access to nutrients, effectively preventing its propagation in the body. And if we can quantify this, we can use that information to kind of better bee husbandry practices to ultimately try and combat colony collapse disorder. So because of this, my question was, how quickly are enzymatic immune pathways activated in Apis mellifera in response to simulated pathogenic threats, and when does immune activity begin to fall? So in order to investigate this, we first used test tubes to collect Italian honeybees from the Langstroth-style colonies on St. Edward's University campus. We only collected foraging bees, and this was because bees show age polyphenism. What that means is that their role in the colony is determined by their age. So in order to standardize age, we only collected foraging bees. And we collected them using test tubes and then returned them to the lab and placed them in the incubator to maintain their body temperature before applying our treatment. So our feeding, we have two groups here. We have our basal and our activated immunity groups. And the function of our basal group is kind of to establish a baseline for what the immune activity looks like without any kind of immune stress. So within that, we had two groups. We had one which was not fed anything upon return and then immediately processed. And then we have our other group that was fed 1 microliter of just pure nectar, which is sugar and water. And then in our activated immunity group, we fed all of them a microliter of 1 mg per mil LPS. And what LPS stands for is lipopolysaccharide, which is a macromolecule component of the gram-negative bacterial cell wall. And so what that does is the immune system uses it to identify and target pathogens. So by feeding them this, we can kind of mimic what an infection would look like without actually infecting them. So in that kind of group, we have groups from 0 to 240 minutes every 15 minutes. And so then upon being fed the LPS, they were then incubated for their respective time points and then refrigerated to anesthetize them before processing. So then the thoraxes were isolated by dissection because that's where the protein content is. And then upon placing the thoraxes in buffer solution, we ground them up with mortar and pestle. And then they underwent several rounds of homogenization and centrifugation before transferring the supernatant, which would have become a pure hemolymph solution. And then using that hemolymph solution, we ran 2 colorimetric immune assays, the first of which was a Red 660 assay, which we used to determine the protein concentration. So using that assay's data and a protein standard, we used a regression to transform the optical density values into mg per mil of protein. And then we ran a PPO assay in order to measure their ability to perform melanin synthesis. So we tracked the change in absorbance over 10 minutes by subtracting the average optical density at 10 from the average optical density at 20, and then took that value and divided it by the total protein concentration in order to normalize in case maybe 1B was larger and therefore had more protein. So these are my results, and I'll first orient you to my axes. So on my x-axis, I have all of my groups, my basal and my activated, and then sorted into their 15-minute time points. And I want to just draw your attention to the fact that it only goes up to 135 minutes. That's because we only ran assays on our samples up to 135 minutes, but we have samples up until 240. And then on my y-axis, I have prophenol oxidase activity. So what that's going to look like is the larger bars are going to be representative of more activity and the smaller of less. So in this red box here, my basal groups, essentially what this indicates to me is that there was a little bit of baseline immune activity just due to handling and stress from being taken out of their colony and brought into the lab, but nothing kind of out of the ordinary here. And then immediately upon the introduction of the simulated pathogen, we see a huge spike of prophenol oxidase activity, which kind of indicates that they are very quick to respond to the introduction of the infection. And so upon that upregulation and the use of the PPO, we then see these decreasing levels, kind of cyclical here, which is indicative of them using up their PPO and also trying to manage their energetic stress. So as they use their PPO, they are kind of down-regulating their expression of that. And then here we see a huge spike again at 105 minutes, which is essentially them trying to ensure that they have fully cleared out the infection, simulated infection, and then down-regulating again at 120 minutes to manage their energetic stress because PPO production is incredibly energetically expensive. So while they're trying to make sure that they are no longer quote-unquote infected, they're also trying to make sure that they don't kill themselves, trying to fight it off. So we see up and down regulation as they kind of get tired but aren't sure if they have completely responded to the infection or not. And then this figure is going to be my percent mortality figure. So on my x-axis, I have all of my time points up to 240. And then on my y-axis, I have percent mortality. And what you see here is starting at around 120 minutes, the amount of bees that were dying increased drastically, which is indicative of the fact that at around the 2-hour mark, they start getting so tired and using so much energy that they've completely depleted their reserves, which ends up being essentially lethal to them. So to kind of just recap and sum up all of that information I just gave you, we saw some basal immune activity just from stress, and then immediately their immune system responds very quickly to the introduction of the simulated infection. And then as the PPO is used, we see it kind of decreasing, and then once they kind of get tired and are trying to make sure that they have cleared the infection, we see kind of cyclical up and down regulation. And then essentially around 120 minutes is when we start seeing some spikes in their mortality, indicating that the energetic tax eventually will claim some individuals who have lower amounts of energy in their reserves. So what I took from this is that the melanin synthesis cascade is extremely quick to respond, but so energetically taxing that production has to be cyclical in order to make sure that they don't essentially deplete themselves of all of their energy. These are my references, and any questions?
Few minutes for questions. I'm trying to think of how to say this. We don't know what causes clone collapse disorder. It's being abducted by aliens or something.
Right.
That's weird. But I'm wondering, like, butterflies, bees that are left, could you look to see if their immune systems have been, like, I'm trying to connect what you just talked about, the colony collapse, and how that can help you figuring out what's going on.
I don't know if I understand the question.
Well, I'm wondering if, like, the bees that are still there after the colony collapse, after the workers are all gone, could you take them and measure their PPO activity and see if it looks like it has spiked?
Yeah, I'm sure that you could, but
Like, are they being stressed?
I'm sure that
They are.
afterwards, yes.
But, yeah, I don't know. I'm just wondering if I can help you kind of understand what's happening with colony collapse.
Yeah, I think that it could potentially, but I'm not entirely sure. But I imagine that we would just find that they somehow were resistant in some way.
Yeah.
This is a very technical question about
Oh my God.
the cyclical nature. So I'm assuming you killed B. You don't get a time course for one bee. So each time point is different bees. So can we really know that it's cycling and not just that oh yeah, some bees peaked at this time and some bees peaked at this time and none of the bees happened to peak in the middle? And can you think of any way to like anything else we could look at that would help elucidate that? Do you have behavioral information that you're just oh yes, they were all
Yeah.
experiencing these surges?
Well, I think that because we standardize the age and they're all coming from the same colony, it's kind of as close as we can get to using the same bee for all of the time points. But as far as finding any kind of behavioral indication of that, the one thing that we noticed in the lab when we were trying to feed the bees was that some of them were way more hyperactive than others. Others were maybe kind of lazy, didn't really want to eat. So there's potential that these two individuals have different amounts of energy in their reserves. So it is possible that could be.
And just be a little bit better.
Yeah.
You guys are really diligent on making sure they consume the whole microliter.
Yeah, any bees that didn't consume the entire microliter were discarded. So unless we saw them actually drinking all of it, we didn't use them.
Can I look at your immune assay graph?
Yeah. This one?
So I'm assuming that at 120 minutes, if a bee was dead, you didn't sample that bee. You only sampled the last piece
Yeah.
when you were in this assay.
Mm-hmm.
And
If
so
they died, we
what
discarded.
differences in when bees die, is there a way to somehow include that as, well, we think that the immune system was really responding, and that's why the bee died?
Mm-hmm.
I mean, it's that the assumption is that it's an over response to the immune system, and so it seems like the death of the bee would also be an indicator that the immune response is
Yeah.
is over.
Yes, for sure.
So I wonder if there's a way to have a graph that combines the deaths with this to indicate that.
Yeah. I'm sure that would, maybe I'll do that next
Just
time.
to try to, just after you do it, just to say, okay, yeah.
Mm-hmm. Other questions?
I have a question. So you use LPS. Have you thought about using any other pathogen-associated molecules to try to stimulate immune responses to see if they respond differently to different viruses versus bacteria versus?
Mm-hmm. We didn't do that, but that would, I think that is kind of like a future direction that we're like interested in.
Thank you.
Our honors mentor up to give her cord, and so that she'll wear during her honors, during graduation as an honor student, and ask me to say a few things about it.
Yeah, so Ellie has been in my lab for about a year now, a little over a year, and I'm extremely proud of her work. We produced one figure so far, but we have a lot of work went into that. We actually have, including myself, 4 people working on the project, and we still have to get to 240 for the one variable, and then we have multiple assays to run after that. So Ellie really has kind of taken the lead on a project that will most likely take me probably 3 to 5 years to complete, and probably will include over a dozen people working on it. It's actually the largest data set I had ever produced. And Ellie and her co-authors have done an absolutely phenomenal job on it. I kind of like to let my students have a little bit of free rein where I give them the objectives and then I let them create kind of the infrastructure. And they were so quick on the uptake, it was ridiculous. And so much so that I asked them to type up all of their, like the way that they kept timing and tracked your bees in the fridge, and you're dissecting, and they're all over the place, right? They did such a good job that I was like, I don't think I can do a better job. I need you to write that down for me because it was amazing. So really, really excited to see what the future holds for Ellie.